decided to build our own in the company around the rapid drug discovery pipeline, and we tried it out and it kind of worked, but our compounds are still going through testing, so we don't know if it's really worked, but it kind of works conceptually, and we think a completely different way of thinking about drug discovery based around knowledge graphs and data science is actually the way to go. And that's probably enough about me.
Thank you so much, David. Really great stuff. And you also talked about some of these. Actually, the first question I'm going to ask after is something you brought up. So thank you for prompting that. And last but certainly not least, Tudor, I promise you, just because I was going to put you first, I did not on purpose put you last. This is just a random order that I generated beforehand. But it's my pleasure to introduce our last panelist, Professor Apria, who I actually know from my work in illuminating the druggable genome. So it's great to see you again. He's a professor at the University of New Mexico, and I believe the chief of the Division of Translational Informatics, working in chemoinformatics, drug discovery, drug repurposing, et cetera. So really grateful that you're here. And please, if I believe you have slides, so please take it away.
Well, initially I was gonna not share the slides considering that no one did, but then David did, so I figured I will do that as well and just riff on the slides instead of just rehashing them. Let me see if I can. I assume you can see the slides.
Yep.
Is that correct?
Mm-hmm.
All right. So I guess the problem that I grapple with, and I really appreciate everyone's thoughts, and the previous panel was fantastic. One of the problems that is rarely mentioned is the veracity of the data. And how do you deal with, for example, when you look at expression data and GTEx says it's expressed and Human Protein Atlas says it's not, or that sort of thing, where you can't really choose sides, but you have to kind of make an assumption that you give priority to one method over the other. So that's one thing, veracity of data. And the other thing that people rarely think about is that even the strictest knowledge has an expiration date. Think if you want Newton versus Einstein, but more recently, the adrenergic receptors in 1948, we thought there were 2. Now we know there's 9 at least. And there's also something called biological drift. Propranolol, which was discovered in early 1960, the published value from 1960 was a PKD of 7.5. And now we know it binds to 3 different receptors with different affinities. So all these things influence how we build our models. And so that's my riff on this slide. But essentially, we have built a knowledge graph system. We are far beyond the capabilities of AbbVie and GSK. We're just dealing with public domain data. But even public domain data takes a lot of effort to stitch together. And what we try to do is take data that are complementary. So as much as we like to keep adding more expression data, we're kind of putting the brakes on that, and we're trying to go towards more phenotype associations, pathways, protein-protein interactions, that sort of stuff. For example, what's not listed here is the latest addition to our database is something called p-hipster. That's a human virus protein atlas that was published by, I'm drawing a blank, Gorka Lasso. He was first author. It was in Cell in November 2019. And of course, given the recent pandemic, it's quite relevant to try to mine not just protein-protein interactions for human data, but also virus-human protein interactions. So once we have all this sort of data, we of course try to feed it into a machine learning model. And just very briefly, we looked at multiple ways to validate this. But in this particular case, we used an external set, which was text mine data from Lars Jensen's diseases database. We are lucky that Lars Jensen is funded through our initiative, so the diseases database is funded through our work. And we used that to make sure that we're not walking into the woods. And we then used 3 different experimental methods to validate this Alzheimer's disease model, which I will not go into, but anyone who's interested, please contact me and I'll give you additional details. But at the end of the day, out of the 20 genes that were predicted by the machine learning model, 3 we are confident enough to say that these are certainly associated with Alzheimer's disease. And 2 of them actually relate to immune pathways, which kind of indicates that perhaps the most recent theory, which is Alzheimer's is actually a sequelae of infection, may be true. And some people in the audience already know, but Joel Dudley was part of that Mount Sinai paper where they looked at patients from Mount Sinai and identified herpes virus into samples from those patients. So there's my link with Joel. Anyone who's interested in the data and mining it, pharos.nih.gov, please feel free to explore and of course give us feedback if things don't work, or if they work, what you like, what you don't like. We would appreciate that. So I'll stop here and let's have the Q&A session.
Wonderful. Thank you, Tudor. And you know, I wanted to save myself for last, the least important member of the panel here, but I just want to at least quickly introduce myself to give everyone a sense of how I fit into all this. So I am a junior faculty member at Mount Sinai. Recently came back from a postdoc at UCSF. And my work is mainly in electronic health records. And it's very interesting because I'm not a computer scientist by training, and I started with the more traditional biostatistical and traditional machine learning models, slowly moving into deep learning. And it's interesting because I've been learning and evolving on the complexity of the modeling strategies. And when I was first introduced to knowledge graphs and graph models in particular, it kind of blew my mind wide open in terms of the possibilities. And now with the amount of data and the computing power and GPU power, that it's all becoming really possible. And I have a student who presented that Ying and I co-mentor, Tingyi, who is really involved in this. So I'm really grateful to be moderating this because it's also a way for me to learn as well. And just before we get into the Q&A session, I just want to quickly ask if there was anyone from the audience that had any questions for the speakers. I see there was nothing in the chat, but I just want to give someone a chance to speak up if they had a question for someone in particular. Okay, everything must have been answered during the presentation. So I have a few questions that I first want to ask everyone. I think we do. You Fu, do you want to ask your questions? So these I think are a little bit more specific rather than general to the audience. Do you mind if we hold off just a little bit until I get into the question section, and then I could add yours in as part of the queue if that's okay? Okay, great. So first, we have so many different domains here, working with clinical data, working with text data, working with image data, working with, in the realm of drug discovery, drug repurposing, et cetera. I just want to first ask a pretty general question for everyone. It's my feeling, I'm not coming purely from the computer science world, more from the clinical informatics world, that, reviewers and I guess just general non-specialists are becoming more and more comfortable and familiar with machine learning, and we're seeing it more in the papers, for better or worse. But knowledge graphs and graph models are a little bit a next step. And my question is, I know that some of us work in computer science departments where it's the language, but I know that a lot of you have collaborators who are not computer scientists or work in companies and have to, you know, report to C-suite execs or even customers or patients that may not be familiar with it. So my question is, how effective, what's your impression on how receptive people are to these models, especially, you know, I'm interested in the healthcare space. So, you know, when we're talking about either with physicians or drug developers, how receptive are people generally of these models? And just to start, I think we could go alphabetically now. Brad, if you would like to kick us off, that would be great.
Sure. I think in general, you know, what we see is that people are receptive to using, you know, graph neural network techniques, using knowledge graph embeddings, when they're kind of led by the potential benefit or they're looking at new ways to solve the problem. I think that there's still a lot of usability gaps in terms of, you know, the expertise required to build these models, the pre-work, which is in many cases is the very hard part in terms of bringing the data together and getting it in the right shape and quality to be able to use it. And then on the graph from a vendor perspective, I think it's also very early days about how we enable some of these features within the query languages and graph models, you know, that we have. So for example, you know, within a graph query, if you want to, you know, send data to be used as a training set, or if you want to embed knowledge graph embeddings into your graph model and then execute queries that will utilize those embeddings as part of your evaluation. So I think those things are relatively undefined today.
Great, thank you. Anne, so at GSK, what's been your experience? I know that you know, the various biomedical ontologies in the tech space are very disparate. So I just want to hear what your experience has been like communicating, I guess, the methods and underlying use cases for which they could be used.
Yeah, I mean, I think in general, we're pretty lucky from the standpoint that a lot of our closest collaborators on the science side are from human genetics and functional genomics, where they really, you know, are grounded in bioinformatics techniques. So I think it's less of a leap to make them familiar with the models that we're using. So, I mean, there's that to begin with. But I think also, I don't know, in my experience, I found that these folks are pretty receptive to these models as long as we communicate the results with the appropriate amount of statistical rigor. So, you know, it's okay for us to say, you know, our model took these bits of data as input and it made these, and this was our training set and our evaluation set, and here's what we predicted. I think as long as we very clearly lay out the experimental setup and the data sources and, you know, the methodology, I think people are comfortable with that. And then on top of that, making sure that we communicate very clearly, these are the bounds, these are the confidence intervals, things like that, to bring it back into, you know, the language that they're used to seeing. But yeah, I found people to be pretty receptive within GSK.
Awesome, great. Thank you. So Jonathan, I guess for you can tell two companies' perspectives in this one.
Yeah, so you know, the nice thing about graph is, I think Tom Plaster in the previous panel worded it perfectly, is that, you know, most of our scientists think in graph, they just don't know it. So you know, when you're able to show that the data is being represented in a way that makes sense to the scientists and the answers coming back in a way that makes sense to the scientists, then it can actually be, you know, quite easy to get traction. I think, you know, where maybe that falls down is when you then push an expectation back on the science community to clean their data, to you know, or when you're trying to convince them to believe a path that isn't the one that makes most sense to them. And you know, I think they're the situations when, you know, there's work to do, for sure. But in terms of just general enthusiasm to use these kinds of models, I'd say there's possibly too much of that at the moment, with an unrealistic expectation of what can actually be achieved. So.
I guess that's with all new models and frameworks is the hype, which I think is I can see it as well. So thank you for that perspective, Brian. What about at AbbVie?
Well, I think the biggest thing is just the realization that this technology and the models and the algorithms are basically irrelevant if you can't make the cultural change as an organization around the way of thinking and the desilozation. And so for us, we are less concerned with the technology platform and what we're building. That is one facet of this whole convergence concept, which is much larger and much more critically involves organizational redesign of R&D. It involves organizational redesign of the R&D IT groups to support that. It involves a complete sea change in the ways of thinking around the data and around the analysis processes. And if you can't make the cultural change happen, it won't matter what you do from a graph perspective. It won't matter from your algorithm perspective. Nothing will matter if you can't make that change. So what we've been doing is going to the groups of scientists and saying, what are the cross-functional use cases? What are the areas where you can't do something right now because the clinical organization has that data locked up and your genomics team has their data locked up and your early discovery high throughput screening group has their data? What are the use cases that would bridge those gaps? And we're allowing those use cases to bring the scientists together, and then they bring their data with them. And that's what's driving our initial implementations in this way, because we think that from there, we'll bring the pieces in, and that will give the trust to the scientists to come to the platform and use it in those ways. And so I think in this case, it's the carrot and stick proposition. It's, y'all got to do this, but we're going to make it appetizing for you. Oh, and by the way, you're not going to have to fight for computational resources. You're not going to have to fight a lot of the data cleaning work because we're going to do it for you. You're not going to have to fight a lot of the 80% of data science being all this harmonization and normalization of your data sets, because we're going to give it to you through your help, harmonized and normalized for you to then use. And so you've got the use cases to start with, drive it from that perspective.
That's great. I think it actually relates to Yu's first question here. And I think I'm stretching a little bit the connection, but I 100% agree with you. I guess the only caveat which she brings up is the idea of like protected health information and whether it's even possible to transfer the data. And I know it's not only just PHI, it's also you're dealing with health systems who may not want to give the data, which is, please.
We ran into it. We actually were discussing this today with our legal folks about derivative knowledge versus data. And when you're talking about consent, right? If you're talking about patient consent in a trial, is consent given to utilize derivative knowledge from that data? Because if it is, if you believe that it is, then you can keep the data, the lower level structured data and extracted data from the trials and from literature and other places, and take this to real world data as well. You can extract doctor's notes as long as you don't extract, and this gets into the challenges of the entities and the extraction process, especially from NLP perspectives. If you can extract the assertions, extract the knowledge from it in a way that it is derivative, is it, and this is the question, is it freed from the consent issues? And do you free it from many of the initial concerns by extraction or by extraction? So that's one of the challenges we've been looking at, is when we think about the research, right? The generalized R&D research data and the graph that will support that, can we take that aggregation out of the regulated domain of data in that space and bring it into this open research domain through that process of extraction?
Great, thank you very much. And the patient data, I know, is such a big issue. Probably could have an entire hour-and-a-half panel on just that. And if you're working in federated models or de-identified models, or what's de-identification, It's a fascinating topic and very important as we move forward. So David, I'm interested in your impression here because you had to, you started a company around this and also got an NSF award around this. So how was the communication of it, especially in the early days? Were people receptive or wary?
Yeah, I mean, I think you have to think of three types of problems that you might solve with knowledge graphs. The first is effectively replacing something that exists already. So something like drug target prediction, which is something we do with knowledge graphs. There are already lots of machine learning models for doing drug target prediction, which are already accepted. So when it gets to the scientists, it's really going to boil down to, as we say in England, the proof of the pudding is in the eating. So either you do better predictions or better insights than the other model, or you don't. And if you do better things, then it'll get used. And the second category is the thing that Brian alluded to, is where you're actually trying to extend some of those use cases by crossing some silos in the background. For instance, a good example might be making preclinical tox work better by pulling in data from clinical studies, or vice versa for that matter. And I think that's a little more tentative, but again, it actually resolves into the first bucket, which is the proof of the pudding is in the eating. It either works better than what they've got right now, or it doesn't. And then the third bucket is completely new problems that you're trying to solve, that it's nobody's responsibility to solve right now, but seem to, if they can be solved, be of huge value. So, finding new pairings of completely new mechanisms of action against disease with drugs that may be in a discarded list in a company that could be used, or drug repurposing is another example. Your problem there is to find somebody who actually wants to do that and pay you money to do it. It might be a fantastic public service if you could solve this, but nobody's incentivized to actually solve that problem. So, what we found is that we want to solve problems in the third bucket, but the way that we make progress and we make money is by solving problems in the first two buckets. And then solving problems in the third bucket is us playing around and demonstrating what's possible, and hoping that in 5 years there will be people paid to do this stuff who will buy our experimental things that we're doing now. So that's my view.
Thank you so much for that. So Marinka, you're obviously in a little bit of a special case here, because I know the department that you work in is very receptive to your work and the public, and you have these collaborations with pharma. But so what's your strategy when you kind of communicate your work both in the academic sphere as well as communicating with maybe non-technical partners or other companies, and to showcase why your method works so much better over these traditional ML models?
Okay, so that's a great question. I have a multiple point answer to that. So
please.
First is that of course there is this area of research in biology and medicine where networks are kind of accepted way to go. Right? There is this entire field of network biology, network medicine that has existed for 20 years or so, when it's clear that certain questions, certain data sets are naturally networks. And so people are not even questioning whether networks are the right modeling language or mathematical language. It's kind of assumed that that's indeed the case, right? Signaling network, various interaction networks, genetic interaction networks, drug interaction networks, and so on and so on. So when those problems arise, it's often fairly easy to convince someone that now we can get rid of some of the feature engineering and manual efforts that would need to be put into the project in order to demonstrate that we can develop models much faster, the models are more accurate, fast, and so on. So that's fairly easy because there are already foundations there that say, well, networks are the way to go. We have many great findings were made using them, and now we have much more accurate, better kind of machine learning methodology in the sense that allow us to get predictions more accurately and faster. So, and then there are these other types of problems where I would say that knowledge graphs are often not first order analysis, right? So imagine somebody goes, they have their favorite image data set that perhaps they just got from somewhere or they generate that, and they will first use only image data it might be cancer, tumor slides to predict patient outcomes. Great. And then after they do that and publish that or finish that project or whatever, then they realize, oh, but it would be great if I could bring in drug information, or if I could bring in cell ontology or tissue ontology, and kind of that would be much better because I would be able to generate insights and so on, right? So that's when in the second phase, then people start thinking about, okay, so how could I do that, right? And then they start thinking about knowledge graphs or some form of integration problem or integration platform. So and I see these are two very different usages of networks, right? So sometimes networks are by definition the kind of measurements that are done. So there are some interactions that are being measured. And sometimes knowledge graphs are a way of are an integrative platform, a platform for integrating data. And in that case, they're often second form case. So kind of from computer science perspective, there's lots of hype right now about AI in healthcare and medicine and so on. From the medical side of things or collaborators, I still see lots of enthusiasm. Certainly something that has changed in the last 2 years. It's like, what I remember when I was doing PhD, it was me knocking on the doors of biologists and saying, I have this method, do you have any data for me to try your method on? And nowadays it's kind of reversed. it's like, oh, we have these data sets, can we try and use our models? So that's really exciting, but I think that there are still like lots of problems that need to be addressed, and it was mentioned before, questions relating to usability. It's like we need to kind of come up with some ways that we get rid of sending those Excel spreadsheets from one place to the other, and kind of deciding that the genes will be renamed just so that Excel will not accidentally change them into dates and so on, which was recently done for those if they have followed, right? And really, what are ways to do that? And these are interesting computer science questions, I think, related to active learning, reinforcement learning. I think the speaker from GSK has mentioned some of those questions related to experimentation and wet lab, design of wet lab experiments, but can we do that for other type of problems as well? So that this integration between the AI model and the user is much more smooth. And there is a way for user to provide feedback, which is integrated in the model in order to improve the model, right? Not only for doing for planning experiments in the lab, but for other tasks as well. And these are some of the hurdles that I think prevent using some of these knowledge graph models in the practice still.
Wonderful. Thank you so much, Marinka. And Tudor, I promise you, you are going to be first for the next question as well. But I'd love to hear your insights, building up this humongous, I think it's unprecedented scale, and working very closely with the NIH. What was your experience, building out IDG, but also using in the cheminformatics space these methodologies for drug repurposing, drug discovery, especially surrounding these unknown targets and TDARC, I believe they're called. Just want to hear your experience of how effectively you were able to communicate this both to wet lab biologists and the NIH.
So I think Marinka summarized it really well. Just a few years ago, it was the other way around. We were like, hey, we've done, I remember in 2004, we had done a virtual screen for a unknown receptor at the time called GPR30. And I went to the biologist and said, look, we've done this virtual screen. Maybe you should test these compounds first. And he didn't even want to touch that for about 6 months. And eventually we came up with a probe that today it's got an IND and it's in clinical trials for melanoma. So that stuff works. But more recently, people are basically, knocking on my virtual door via emails and texts to say, hey, can we work with you? And this goes back to something that Bill Egan, who used to work at Pharmacia, he called it the rule of one in computational chemistry, which was opposite to the rule of five. Some of you might know that. You're allowed to make one mistake. This is not the case anymore. People are becoming more tolerant to failure of prediction because they understand that biology is uncertain. So I think in that sense, we are seeing a paradigm shift. It's pretty much convincing, and I'm almost convinced to the fact that AI may actually succeed in drug discovery. I'm not there yet because I'm still an informed realist, which is the opposite of an optimist, if I could say it that way. So with respect to drug repositioning, I guess what's unprecedented today is the availability of data that comes from multiple sources, which allows us to integrate not just biological data, but also clinical data. So you have access to, NIH has these wonderful tools, CleanGen, CleanVar, you know, that there's a lot of data that is becoming more and more reliable, and it's got that clinician curated label that allows us to put more weight. So even though we can't ascertain what's true and what's false, we can at least come up with a scoring scheme that allows us to weight the data based on some sort of credibility score. And I try not to call it a credibility score, we call it confidence, but it's pretty much the same thing. So we try to implement assertions based on that. And we're also trying to perturb. So one trick that I realized early on we have to do is change the seed, change the code. So if you have a code that runs in R, run the same code in Python and see if you get the same results. If you get very different results, probably the models aren't as reliable as you think they are. So try to implement some perturbations to the model, change a little bit the negatives and the positive labels, shift them just a tad and see whether you get the same trends. And if you get the same trends, you might begin to start trusting. Like in the top 200, you see roughly the same, just slightly reshuffled, then you have a model. If it's a totally radical shift, then you probably don't have a model. It's all random.
That's great. And I think that very much relates to the second question that you had, and that it was one of the questions that I had. So maybe I can actually just quickly follow up with you first on this. Is okay. She asked, or you asked about about the uncertainty of these things, and actually the quote you said about not being penalized for not getting perfect predictions. I wish I could go back in time to a reviewer on one of my papers and have that quote there. Right. But you was asking, is there a way to test for significance, or the best way to test for significance, and the idea of causality? I know in a lot of work that especially works on electronic health records, it's a very touchy topic. So I'm wondering if you could just give a few sentence answer. And I realize we're getting close on time here, so I want to make sure a few people get to answer. But if you can give your thoughts, that would be great.
So one advantage on my side is that I'm embedded within the Department of Medicine, and I basically I used to sit at the same table with division chiefs from just all the disciplines in internal medicine. And I would often come up with, or they would often come up with a problem, and then I would just quietly go and run a model and come up with some predictions. And they would sometimes be mind blown, because initially they were like, no, this doesn't make sense. And then if you dig, it turns out that it does. I'll just give an example, glucagon in asthma. And everyone was, no way. And it turns out that glucagon has a direct bronchodilator effect. I didn't know that. The model somehow found that out. So this kind of little details ended up giving me more and more credibility. So by the time we said, look, you can't just test one gene, you have to test 20 genes. Well, we don't have the funding. I'm like, let's just find the money. And also it's not that difficult to run 20 experiments instead of one, because at the end of the day, it's all about the ability to take the science where it gets you. Causality is very much on my mind. I wish I could tell you that there is an easy answer to that. There isn't. Part of the problem is that biology keeps changing. We scientists have a lot of biases, and those biases tend to push us to record certain observations where we tend to discard others. And so sometimes it's even better to just take the supplementary files and process them yourselves. So that's what we do. You probably know Avi Bayan and Mount Sinai does that as well.
Of course, yeah.
So we do a lot of that work where we actually try to avoid bias as much as possible when we evaluate the data. Great,
thank you. And again, I was so interested in all these questions. I probably should have kept a tighter time schedule here. We're getting close to the end here. I do have a few questions that are really I've been really hoping to ask. I think I went a little overboard with how many I planned, but I just want to again give the opportunity to the audience to ask any questions.
Well, there's questions that come in, and I just got one comment.
Oh, please. Yes, sorry.
Confidence, certainty. You know, I think you know, a lot of the graphs we work with, you know, are in a large part built from the scientific literature. And it doesn't matter how good our NLP gets, you've got to recognize the weaknesses with that as a data source. Generally, you know, I think it's been mentioned several times, you know, the lack of the capture of negative data in there. But I think also sometimes the way that we typically weight our confidence in an assertion coming out of the literature doesn't really reflect the way we would do it manually if we interrogated that paper. If we interrogate that paper, we look at, well, how many samples did they use? Do I believe the antibody was a good quality one that they used to take that measurement? You know, what was the diversity of the patient population? These kinds of things. Whereas, you know, do I believe, you know, the credibility of that lab? These kinds of questions, right? Whereas when we're you know, trying to draw this information out through computational means, we're looking at the quality of the journal, and you know, I think we need to get better at actually capturing the quality metrics that we as scientists would use to measure whether we believe, to assess whether we believe a measurement or not.
I'm going to quickly comment on that, because we try to look at the impact factor as a way to bias the confidence in information. Impact factor did not help.
I was going to say, it calls out one of the actual more interesting use cases, and something that we've already started to see even at the fledgling state of our graph is when you bring in all of your assays, all of your high throughput screening and all of your preclinical assays, you will find conflicts. You will find places where knowledge assertions from literature directly conflict with what you have assay and testing data for. And I think that's one of the huge opportunities of taking this point of view, is because you can, when you find those conflicts, there's two paths. Either your assay is busted and you better go back and figure out what's wrong with your testing and your assays, or there's something novel about what you're looking at and the difference between what your metadata that you have around the way your assay is being conducted, or the environment or something about that assay, and what has been presented in the existing literature. And both of those are opportunities to help inform general knowledge and understanding about biology in this case.
Most, I mean, my experience at AstraZeneca, you know, most of what was done in the preclinical space was to disprove the literature. But you then don't capture that. You then don't publish that information. So everyone else has to go and disprove it themselves, you know, so yeah.
Yeah, if I can also add a comment from the ML perspective, right? So recently, there's been this lots of enthusiasm about deep learning for graphs and graph neural networks and so on. And in the first few years, everyone was more or less excited just to beat the AUC scores, right? And now people have started asking, including us, right? Really, how sensitive are these predictions made by these models to small perturbations in our knowledge graph? So imagine like, oh, well, if I take a particular entity and I add a few edges or remove a few edges, which would translate to a few additional relationships being extracted or not extracted from literature, for example, how would that affect prediction made by the model, right? So it turns out that, well, more or less all models that are state-of-the-art models nowadays for ML are extremely sensitive to these perturbations even though perturbations might be done at random and are sensitive to perturbations in graph structure, in node features, all kinds of things, let alone adversarial perturbations. Imagine somebody would come and would intentionally try to perturb your knowledge graph to change the prediction, which might not be a huge problem for some of the basic science research, but it actually might be a very relevant problem for more translational clinical applications, either to perhaps, because it's possible nowadays to think about, well, it's possible to define optimization tests to say, what are the minimal change to the graph that I should make such that that will change the billing code for a patient, right? And it's possible to do that nowadays, right? So this whole question appears about how can we train these models robustly? Like, and often people use this word, oh, robustness. Of course, we want robustness models to be robust. We use them very naively, or just like throw these words around. But it's really an important challenge how to train these models robustly, because the data that we have is missing in so many different ways, confounded, contradictory, and so on and so on.
This discussion is really good. I mean, particularly that there was one of the questions I don't think we're going to get to, but that to me is a fascinating topic. You know, we have missingness not at random. We have, you know, we're learning so much about the you know, ethnic and racial biases that a lot of these ML models. I mean, there was one, I think insurance companies were using them. I mean, these are devastating things that really impact lives. I think, again, that's a humongous topic that I love to talk further about. But in the auspices of time here, I have a little bit of a selfish question that we'll start with Anne about, and it's, you know, for a lot, there's so many different types of data that we're dealing with, right? And myself especially, even within the patient health data, right? We have imaging, we have notes, we have structured data. So what are your strategies, and Anne, we'll start with you, about, and even within different corpora of, like, you know, you have prescription labels, you have physician notes, you have nursing notes, which differ a little bit, you have radiology reports that differ a little bit. How do you effectively harmonize them? And especially, you know, within any kind of analytical framework that you're doing, whether it's machine learning, whether it's just simple, you know, creating models separately and joining them together, you know, what are your effective strategies for harmonizing and or jointly learning? And, you know, as a side question, if you prefer to answer the other one, you know, all these data sets have different update frequencies and being depreciated. So if you could, in the auspices of time, maybe give a 1 minute answer, if that's possible, or 1 to 2 minute answer.
Yeah, sure. So, I mean, the first thing to do is to make sure that you're pulling in the data that's relevant for the question you're trying to answer. So you know, we don't want to pull in a data set that's not relevant, obviously, to the question that we're building the model for. But I mean, we make big use of ontologies. So we try to map everything to common ontologies wherever possible, and use inter-ontology links to make sure that we have, you know, one representation of each concept to the greatest extent possible. That helps. But I think then also, you know, we try to come up with sort of small ground truth data sets wherever possible, just common machine learning practice. But we use those to evaluate the robustness of our model. Rinka, to use your term again. So, you know, we systematically or at random will drop edges from the graph and retrain. We'll systematically add edges, or at random, we'll change seeds and things like that. And then look at how stable the predictions are on that ground truth data set as we do those things. And you know, we decide to trust things when we're getting, you know, as Tudor mentioned, you know, getting sort of consistent results, even though we're making these changes to the underlying graph.
Thank you, Anne. So I want to also give Brad a chance to comment on either of those, but either the harmonization or the operationalizing of pulling in different public data sets and different ontologies that have different update frequencies, whatever you prefer to comment on.
Yeah, I mean, we certainly see customers doing it. You know, there's lots of different implications that people have in term from a platform perspective. You know, time to live features for automatically deleting pieces. You know, one of the other things that we see customers doing is, you know, building catalogs of the data sources with these kinds of metadata attributes, so that they can, you know, very quickly understand within all of the data sets that they're managing what those update rates are. Use those as input into some of their model making processes.
Great, thank you. So David, I know some of the stuff you built takes a whole bunch of public data and you know, even literature that updates constantly with new articles. So what's been your strategy to effectively deal with like a living beast?
If I'm honest, I think our best strategy is we've got some super high value, talented people in the company with decades of literally, I think we've got over 100 years combined experience in this field who understand the biology, understand the chemistry, understand the science, and understand the data and machine learning and the data science. And they put a huge amount of grunt work in. Of course, you know, one of the things nobody likes to admit about all data science is you know, it's 90% data wrangling and cleaning and mapping, and it's 10% doing the cool stuff on top of it. So I think my answer is we've hired some really talented, fantastic people who build very complex, subtle scripts and workflows to manage all this stuff. I mean, we do have all the ontology mapping. We use ontologies where we can. But this is complex stuff. I mean, it's the human body. We in our, you know, even if everything's perfect, it's all going to be rubbish because it's just a very poor, crude model of what's really going on in our bodies anyway. So, so it's right that it's complicated, and you have to work hard to deal with that.
Thank you. I'm glad the AutoML stuff hasn't put us out of business yet. It seems a lot of the people are still important here. In the auspices of time, I do want to ask one last question, and we'll start with Tudor. What is the most exciting thing to you right now about the future of either knowledge graphs or graph models? I'm particularly interested in evolving graph models. I know Marinka has a lot of work there. So if you could just give maybe a 30-second comment if possible.
I think the ability to take real-time data that's been deposited, say, by the groups of Nevan Krogan, who did that in March with the mass proteomic pull down for the virus. That sort of research, if it's done early and frequently enough, then it can lead to tremendous advances in therapeutic discovery. So that's what I'm excited about.
Cool. My good friend was actually co-first author on that paper. He's in Krogan's lab. So so Brian, if you could just very quickly, what just you're most excited about? What's cool? What's upcoming? What's
Sure. So I'll give you two answers really quick, because there's the what's exciting and then what I think is actually going to be tangible and real and valuable. To me, what's like fascinating and exciting is the interplay between the quantum computing space and the graph algorithm space. As we start to look at the opportunities for, you know, quantum scale graph optimization algorithms and what's going on there, it's just fascinating. Like the math is insane, the technology is insane. It's the Wild West, right? It's our chance to relive computing the way it was in the 50s and 60s. Where you just have this fundamentally new thing coming along. So that's exciting to me. Now, from a value proposition space, and what I think is really going to be valuable is when we start to bring the real world data out of the world of big data and into the world of graph data in a really true and valuable sense. And that is a challenging prospect for a lot of the natural language type things when we start thinking about EHR and medical records and how do we parse what's important from there into knowledge. But I think if we can bridge the research to real world data gap, the complexities, if we can get through that I think is just the fascinating opportunity to break the pipeline, to take this stage by stage siloed pipeline, just break that whole concept.
Cool. And I see we're at time here, but the question was asked. So Marinka, please just finish us off. Last comment here for the conference. What's cool? And I've read a lot of your recent papers, so there's a lot that's cool in your mind. But what's the next thing? And I'll take notes.
Sure. I really liked what Brian said about bridging this gap between basic research science data and patient data. I think that's kind of very cool from the data point of view. From the methods point of view, I'm most excited about this notion of auto-distribution generalization, or kind of thinking about never before seen phenomenon. Either there are no new diseases or very rare diseases, new molecules that are not yet drugs. And where I see here the roles of graphs and knowledge graphs is that they allow us to jump across these data contexts, to kind of relate something that is very new and there's nothing in its surroundings, so we cannot use these guilty by association principles that we have used for 20 years. Just because something is structurally similar, it will have the same role or whatever. But now graphs allow us to build these long meta paths or data contexts, however you want to call them, and all of a sudden relate this unknown, previously unknown entity. Might be COVID-19, it would be some molecule that is not yet drug. We can relate it to the knowledge that we already have or know something about. And that's something that I'm very excited about.
Yeah, that was one of the things I wish we could have mentioned was COVID in the context of knowledge graph. And I know Ying and many others have work here, but we're 1 minute out after the hour, and I want to make sure those of us on the East Coast get to have the rest of our night. I want to thank you so much to the panelists. This was really. I wish it could go on for another 2 hours. Hopefully, we can maybe continue some of this conversation on Twitter. I want to very much thank Ying for really organizing this entire thing. I mean, it's her brainchild and all of her effort that really went to this. So thank you so much, Ying, for inviting me and inviting everyone to take part. And I'll leave the closing remarks to you.
Okay, Ben, thank you so much for this wonderful panel. We all learned a lot. We're also looking forward to our new future. So, and I want to thank all these people. We still have 35 people in this room. I want you to thank you to stay the whole day with us and 8 hours. And hopefully we know a lot about knowledge graph. Hopefully you know how to apply knowledge graph in the future. Thank you very much. Enjoy your evening. Okay.
Thank you.
Thank you, everyone.
Bye.
Thanks, everyone.
Bye.
Thank you.